Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Okay, let's unpack this. Imagine a cancer treatment that you inject in just one spot, but then it goes to work all over your body, even making distant tumors disappear. Sounds almost too good to be true, right? Well, today we're taking a deep dive into some new research that suggests this might just be, a reality for aggressive cancers. Our focus today is a really compelling breakthrough. It's in a specific class of cancer drugs called CD40 agonist antibodies. Now, for years, these drugs showed huge promise in the lab, especially for activating the immune system to fight cancer. But here's the twist. They consistently struggled in human trials, often causing, severe toxicity. So our mission in this deep dive is to explore how scientists engineered a new drug, it's named 2141V11, to overcome these exact challenges. We'll unpack the really remarkable results from its phase I clinical trial and talk about what this could mean for the future of treating some of the toughest cancers out there.
Yeah, and what's fascinating here is really understanding the history. These CD40 agonist antibodies, for 20 years, they were a bit of a paradox. They looked incredibly promising in lab tests, but then were kind of a consistent letdown in human trials. In animal models, these drugs were highly effective. They prompted the immune system to kill cancer cells, making them a really exciting area of research.
Okay, so if they work so well in animals, what were the big hurdles? What stopped them from working well in people?
Well, the major setback wasn't just, their limited impact on the actual cancer in patients. It was the severe systemic side effects. We're talking about really dangerous inflammatory responses all through the body, very low platelet counts, liver toxicity, even at low doses. Basically, the body has these CD40 receptors pretty much everywhere. So when the drug was given intravenously, systemically, it caused too broad a reaction. It triggered an immune response everywhere, not just where you wanted it at the cancer.
That sounds like a huge problem, almost insurmountable. So did researchers eventually find a new angle, something that let them move past these issues?
Exactly. Something fundamental had to change, and it did. In 2018, the Jeffrey V. Revetch lab at Rockefeller University, they engineered an enhanced version of this antibody. They called it 2141V11. The key innovation was modifying the antibody so it binds much more tightly to the human CD40 receptors. And importantly, it enhances its cross-linking, basically, how it clusters these receptors together. This actually made it about 10 times more powerful at triggering that anti-tumor immune response, at least in the models they used.
Okay, that's interesting. But if they made it more powerful, how on earth did they avoid making the toxicity worse? Was there a completely different approach to actually giving the drug?
Yes, and this is really the crucial shift, the game changer. Instead of giving the drug intravenously, spreading it everywhere and hitting all those receptors, they decided to inject it directly into the tumors. Intratumoral injection.
Okay. Localized.
Exactly. And when they administered it this way in their preclinical models, they saw only mild toxicity because the action was primarily kept right there at the tumor site.
Getting the dose right for people must have been tricky then, especially after the earlier problems. How did they figure that out? What models did they rely on?
Yeah, it was absolutely crucial to get this right. Interestingly, studies in non-human primates, like macaques, they didn't really predict the human response accurately. Turns out the drug's enhanced power relies on a specific human immune component, a receptor called the Fc gamma receptor, which macaques don't have in the same way.
So primate studies weren't the best guide here.
Right. So they really relied on humanized mouse models. These models mimic the relevant human immune pathways much better. And those models were essential for estimating a relevant dose. They allowed for, a very careful selection of a really low starting dose for the human trials, minimizing the risk.
That careful groundwork seems to have paid off because the results from the phase I trial of 2141V11 sound pretty remarkable. What was the main goal of that first trial, and who were the patients?
So the primary goal for this phase I trial was, first and foremost, to find a safe starting dose for clinical use, and also to start understanding how the drug actually works in humans. The trial included 12 patients. They had various aggressive metastatic cancers, things like melanoma, renal cell carcinoma, different types of breast cancer, and these weren't patients new to treatment. On average, they'd already been through a median of 5 previous treatments, so these were really tough cases where other things hadn't worked.
Okay, and the big question, safety. That was the major stumbling block before.
Yeah.
How did 2141V11 do, especially being, more potent?
This is where it really shines.
Yeah.
The safety profile was, frankly, unprecedented for this class of drug. None of the 12 patients experienced those severe side effects that plagued the earlier CD40 drugs. There were no dose-limiting toxicities observed. That means they didn't even reach the maximum dose people could tolerate. Yeah. The most common adverse events were just mild to moderate. Things like fever, chills, some reactions at the injection site, mostly seen at the higher doses, and crucially, no significant drops in platelet counts, which are vital for clotting, or increases in liver enzymes, which indicate liver damage. Those were the big problems before.
Okay.
Now, 3 patients did have serious adverse events during the trial, but importantly, none were considered related to the drug itself.
That safety profile alone sounds like a massive step forward. But of course, safety isn't enough. Did it actually work? Did it fight the cancer?
Yes, it absolutely showed impactful efficacy. 6 out of the 12 patients saw their tumors shrink. Now remember, this is a small group, and these patients were heavily pretreated. So seeing that kind of response rate, 50%, is really meaningful. These were patients running out of options.
And this next part is just incredible. 2 of those patients, right? One with melanoma, one with breast cancer. They actually went into complete remission. Their cancers vanished entirely. That's the ultimate goal. Tell us about them.
Yeah, the stories are really compelling. One patient, an 89-year-old woman with aggressive melanoma. She had dozens of tumors all over her leg and foot. But they only injected 1 tumor on her thigh. Just one.
Just one spot?
Just one. After multiple injections into that single tumor, all the other tumors, the ones they didn't inject, they disappeared. And she stayed in remission for an incredible 28 months. That's a very long time for that kind of cancer.
That's amazing. And the breast cancer patient, also a notoriously difficult cancer, especially when it's spread.
Yes. The second case was a 67-year-old woman with aggressive stage YV breast cancer. She had tumors in her skin, her liver, and her lung. Again, they only injected the skin tumor.
Just the skin one.
Right.
Yeah.
And yet all her other tumors, the ones in her liver, the ones in her lung, they disappeared too. Even her tumor markers in her blood went back to normal.
Okay, this effect where you treat locally, but tumors far away also respond. There's a name for that, isn't there, in oncology?
There is. It's called the Abscopal effect.
Abscopal effect.
Right.
Right, and how significant is seeing that?
It's highly significant to get a systemic response from just a local injection like this, where immune cells activated at that first site travel and destroy tumors elsewhere. That's not something you see very often in, well, any clinical treatment. It's another really dramatic and frankly unexpected result from this trial. And it's crucial because it strongly suggests this local treatment is kicking off a body-wide immune attack.
So how does that work? What's happening inside the body? How does injecting one tumor sort of teach the immune system to hunt down the others?
Well, the research really dug into the immune environment within the tumors. They found that in the patients who responded, their tumors, even the ones they didn't inject but it still shrank, developed these structures called tertiary lymphoid structures, or TLS.
Tertiary lymphoid structures, okay.
Yeah, you can think of them as like little clusters of immune cells.
Yeah.
T cells, B cells, dendritic cells forming right inside the tumor. They actually resemble small kind of temporary lymph nodes.
So the tumor itself becomes like an immune training ground, building its own little headquarters for the fight.
That's a great way to put it. Precisely. And the presence of these TLS structures is known to be strongly associated with a better prognosis, a better response to immunotherapy in general. Finding them even in the non-injected tumors that responded, that really points to this idea that once the immune system gets activated against the cancer cells, those newly trained immune cells migrate out to other tumor sites.
And what about the bigger picture? Was there evidence of immune activation throughout the whole body and not just seeing tumors shrink?
Oh, yes, absolutely. There was clear evidence the whole immune system was getting revved up.
Yeah.
In the patients who had those complete remissions, they saw a big expansion of key cancer-fighting cells. Specifically, CD8 plus T cells. Those are the body's direct cancer killers, and also T follicular helper cells, which kind of help coordinate the attack. And they did T cell receptor analysis, which showed that these crucial CD8 plus T cells were clonally expanding, meaning specific populations of these killer cells were multiplying rapidly and then spreading throughout the body. That directly links the local injection to a systemic effect. They also saw an increase in inflammatory and immune-activating cytokines. These are signaling proteins that ramp up the immune response in the blood, especially at higher doses. So more reinforcement of that systemic activation.
And didn't the researchers use those humanized mouse models again to kind of confirm these mechanisms? What did the mouse studies add to the picture about how these TLS things work?
Yeah, they did. The mouse models confirmed the formation of TLS and also showed that abscopal effect. And connecting this to the bigger picture, the research suggests that these tertiary lymphoid structures within the tumor act as sort of autonomous hubs for activating the immune system. They essentially kickstart the immune response right there at the tumor site, maybe even without needing the traditional lymph nodes for that initial step. And then those activated T cells can travel and hit tumors elsewhere.
Interesting.
And interestingly, while B cells are definitely present in these TLS structures, the mouse study suggested they might not be absolutely required for the direct anti-tumor activity of this specific drug, 2141V11, in that model, though they might help provide a kind of scaffold.
This raises a really key question, right? Generally, only maybe 25–30% of patients respond well to immunotherapy. So the huge challenge is figuring out who will benefit and how we can maybe turn non-responders into responders. The study hinted that something called T-cell clonality might predict response. Can you unpack that a bit?
Yes, that's a potentially crucial insight. The two patients who had those complete remissions, both of them had what's called high T-cell clonality before they even started the study. That means they already had a diverse and strong population of T cells capable of recognizing many different targets on the cancer cells.
So their immune system was kind of already primed in a way.
Exactly. It strongly suggests that there might be existing immune system characteristics or maybe predispositions that are needed for this drug to work really well. Pinpointing those characteristics is a major focus for future research. Understanding this could help us select patients most likely to benefit, and maybe even figure out strategies to prime the immune systems of people who might not otherwise respond.
Okay, so what does this all mean then, big picture, for the future? These results sound incredibly exciting, but it was a phase I study. Small numbers. What are the main limitations we need to keep in mind, and what happens next?
Right, you always have to be cautious with phase I results. The small number of patients, the limited mix of cancer types represented, these are definite limitations. We need more data. But despite those limitations, the really favorable safety profile, combined with those profound clinical responses, complete remissions in heavily pretreated patients, that provides a very strong foundation to keep investigating. This feels like a significant step forward. It pushes the boundaries of what we thought a local therapy could achieve systemically.
And it sounds like that further investigation is already happening.
Oh, absolutely. There are nearly 200 people now enrolled in further studies, phase 1 or phase 2 trials.
Yeah.
They're looking at 2141V11 in other tough-to-treat cancers, like bladder cancer, prostate cancer, even glioblastoma, which is a very aggressive brain cancer.
Right.
These larger studies will be absolutely crucial. They'll help illuminate why some patients respond and others don't. And importantly, how researchers might be able to improve that response rate, maybe by combining this therapy with other treatments to get even better results.
So wrapping up, 2141V11 really looks like a significant step. It demonstrates a safe, potentially very effective new approach. It harnesses the immune system in this unique way, activating local defenses that then ripple out through the whole body. Imagine a future where, one targeted injection doesn't just treat the tumor it touches, but it actually educates your entire immune system to seek out and destroy cancer wherever else it might be hiding. This research really gives us a glimpse into that powerful possibility. How might these local immune factories really transform how we think about cancer treatment down the road? It's a fascinating thought, and we'll definitely keep you updated as this groundbreaking research continues.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.